The cell theory established in the middle of 19th century proposed that all living organisms are composed of cells. Cells can arise from the pre-existing cells. Plants are eukaryotes and their cells are divided into compartments, each of which is enclosed by at least one lipid bilayer membrane. Three distinctive features of the plant cells are the presence of a cell wall composed of cellulose and other polysaccharides, plastids and vacuoles. The components and structure of the cell wall are important aspects of differentiation. Within the wall, the usually pressed up against is the plasma membrane. This membrane regulates the uptake and exit of molecules and ions. Membrane is involved in sensing signals from the environment and other cells in the plant and cellulose synthesis. In addition to the external boundary, plant cells have an internal boundary, the tonoplast, which encloses the vacuole. Expansion of plant cells occurs mainly in the vacuole, so that this is the largest compartment of most mature cells. Most of the soluble reserves of organic molecules, sugars, organic acids and amino compounds are held in the vacuole. The vacuoles contain hydrolytic enzymes, so that some of the waste can be digested and recycled. Because of its dissolved solutes, a vacuole regulates the osmotic properties of plant cells. The organelles and most of the metabolic apparatus of the plant cell exist in the cytoplasm between the plasma membrane and tonoplast. Chloroplasts play a very important role in photosynthesis, and amyloplasts, which are filled with starch granules and occur in storage tissues in seeds, roots and stem. Chromoplasts, which contain yellow or orange carotenoid pigments in solution in lipid droplets and occur in many flowers and plants. Eoplasts, which are juvenile form of plastid that occur in embryos and apical meristems, and etioplasts, which develop in tissues in the dark and lack pigments.